Hi everyone, and thanks so much for coming. Lovely to see a full house again this week. So we have been asked in previous sessions that we would introduce what people are doing in the department, and we have sort of paired up people, senior and more early career researchers together. And so Gigi is, as a professor, is our senior researcher, and is going to talk about some of the work that he's been doing with Harriet Baker, whom some of you may know. He's from oncology and working with us as part of her PhD. And it's lovely to see that collaboration between radiology and oncology, and going to hear more about what you're doing.
Great, thanks, William.
And the ACI, indeed. And over to you, Gigi and Harriet.
Thanks. So, thanks for the introduction. So, we were going to go over sort of the overall radionuclide related research environment and what happens, not necessarily just what happens here on level 5, but the things that are happening throughout the campus that we're involved in. And the more I look at this, the more there's actually a lot of stuff that's going on, a lot of people involved. So hopefully I'll make you understand what this is about and how it works. So I'm going to talk about some of the small project that we've done and it's finished, and then Harriet's going to talk a bit about projects that are ongoing, and then we'll wrap up with a few sort of more general things about everything that happens here. So research in this field in oncology, in radionuclide research, it's all about drugs. So we use radio-labeled drugs for cancer. So we want to do what we can do compared to other techniques. We can specifically target biochemical properties of cancer, and that way we can have uptake of radiopharmaceuticals specific to that biochemical property. Against a low background, and that's what we try to do. So we use these biomarkers that we target for imaging. For imaging, we use to detect, diagnose, or characterize disease, essentially doing sort of in vivo histochemistry, if you want. But also more recently, and that's the thing that we're mostly focusing on, is the aim of getting to something that you could actually then use and exploit to deliver lethal dose radiation to cells. And goal is obviously to have an impact on how we manage disease and to improve outcome in patients. So this field, this sort of theranostic field, has been expanding in the last 20, 30 years. Most of what's happened is focused around these protein coupled receptors. It's these targets that have been utilized mostly because they are easy to develop ligands for, because most of the ligands are actually biologically relevant molecules that can then be adapted to these places. And many of these targets are present in a lot of these cancers. So we're going to talk a little bit about bombacin in the first part of the talk. Bombacin has been around for many years, and there's been approaches at labeling bombacin targeting ligands with many different types of isotopes, so proteins for PET and SPECT. And more recently, we're looking at how to use these for radiolabeling with nuclides that are useful for therapy, such as beta emitters and alpha emitters. So the whole field of radioligand therapy has boomed. There's a lot of industrial interest, big pharma interest. And this is a sort of a map of what's happening. I'm not sure if you can see the entire, all that's there, but we actually have a lot of interactions with many of these companies, and many of those items that are there, we are to some extent involved in at different stages of research to get research started in these areas. So the two targets that are in the middle, which are Lutathera and Pluvicto, these are two products that are owned by Novartis, who target PSMA and somatostatin receptor 2. These are the only two approved drugs, but there's a wide range of research at different stages of development in many different areas, and most of those targets up there are actually G protein coupled receptors. Somatostatin receptors, for example, are G protein coupled receptors. PSMA is actually a cell membrane enzyme, but most of the others that are in development. FAPI is another cell membrane enzyme. It's fibroblast activator proteins present in cancer-associated fibroblasts. But there's other G protein coupled receptors that are being looked at in that wheel that you see up there. So I'm going to talk about what we've done with GRPR. So we, and this again is a Novartis ligand that we've used, and Novartis has developed both a diagnostic and a therapeutic in this area. So we're going to talk a little bit about what we've done in imaging with GRPR. So like I was saying, many years these G protein coupled receptors have been recognized as potential targets. So gastrin-releasing peptide receptor or bombesin receptor are part of a family of 4 receptor subtypes. Bombesin 2 or GRP is the one that we're focusing on here, and it has been found in several tumors. So the paper cited there on the right is a paper from Jean-Claude Royby from 2003, when the boom of somatostatin receptor imaging started, and we were looking at what other targets can we look at. And so there was a lot of work looking at quantitative autoradiography to look at actually the levels of expression of all these receptors in different tumor types. I've highlighted there that the cancers that at the time were looked at in terms of the GRP bombesin receptor expression. And there's some sort of fairly big item cancers like prostate and breast that were highlighted at the time. So we started developing ligands for these a long time ago, and things have evolved during this time. So initially, and actually Bracco had a compound called AMBA, so receptor agonist. So essentially you take the protein, you break it down, you find the binding portion, and you use that as a ligand. But that ligand was an agonist. It was horrible because tiny amounts of the compound actually created very acute side effects. And so it actually bombed pretty much because of that, because it wasn't, you couldn't administer it in quantities that were suitable for use. So people started looking at antagonists. So they identified portions of the receptor, and you can see those diagrams up there, where you essentially did not activate the receptor by interacting with those portions, and design sort of different peptides in order to sort of avoid the agonist effect. So these antagonists were then studied in various forms by different authors, and NeoB here, this was developed by a lady called Theamina, who works in Greece, in Athens, and was then sold to Novartis for development. Initial studies with this were actually in prostate cancer. So the NeoB is the tracer that we have used here, and those are some of the early images that were obtained in prostate cancer. So we're going to talk about GIST, gastrointestinal stromal tumors. There was some evidence that in addition to the tumors that I showed you before, both cell lines obtained from GIST tumors as well as immunohistochemistry study in human tumors show that these GIST tumors could actually overexpress GRPR. So just a bit of a summary about GIST, and that's actually Ruth Casey's pictures, because she's sort of been the clinical counterpart in this imaging part of the study. GIST are rare cancers, 1 in 100,000. It just so happens that Ruth has a GIST clinic on that she follows here, so she gets patients from around the country to see. Most GISTs are so-called mutated GISTs, so where there's a mutation of either the KIT or the PDGFRA gene, the vast majority of these. And these, essentially, the biochemical feature here is that there's constitutive activation of tyrosine kinase. And so these tumors are actually responsive to tyrosine kinase inhibitors. So Glivec, imatinib, is the treatment for these tumors, and these tumors respond very well. But a small percentage of these GISTs are actually so-called wild-type GISTs. So this is a misnomer, because they actually don't have mutations in KIT or PDGFRA, but they actually have other mutations that drive the disease. And Ruth is particularly interested in these succinate dehydrogenase deficient tumors. So since these tumors really have no effective treatment with it, they resist TKI treatment because the actually biochemical driver is different. So we were interested in looking at this as a way to bring a therapeutic strategy for these patients. So we did this initial imaging study in 12 patients, which we recruited. These, as you can see there, these are the table of the characteristics of these patients. So genotype, they all have SDH mutation of some sort, so they're SDH deficient. Most of these were, well, all of these were the primary site of the stomach and were metastatic representation. Most had, well, all of them had liver metastases, and others had other peritoneal metastases. There's a wide range of the sort of how aggressive these tumors are, and I suspect in this population we've mostly captured the ones that are sort of indolent and sort of progress slowly over time. And so part of the reason why this population is also biased, and we'll look at that later when we look at the imaging, is also the fact that these are sort of patients that have been having a tumor growing slowly but steadily for many, many years. So, so this was the population we did. We just did the very simple experiment of looking at PET with NeoB and looking at whether this was effective in imaging disease. So this is the image of one patient. For those of you who aren't familiar with that, this is a MIP image. It's a projection image of the 3D representation of the distribution of the receptor. So the black areas are where there's higher activity, and you can hardly see the outline of this patient, but you can see that pretty much all the extensive liver metastases light up, and you can see that the pancreas also is very avid, and that's physiological activity. That's best seen on these fused images. So you can see the pancreas is extremely high uptake, SUV values of 50, 60. So obviously the pancreas is the site where there's physiological expression, because the gastrin is actually produced in the pancreas. So the gastrin releasing peptide physiologically is released from cells in the gastrointestinal tract and stimulates the pancreas to produce gastrin. So this patient here, as you can see, has very extensive liver disease and also disease in seminal vesicles. And you'll probably spot some areas in the peritoneum as well, which probably corresponds to metastases. So one of the things that came out of this is that the intra and inter-patient variability of infection is very, very high. And I think that's mostly related to the fact that, again, these tumors slowly growing have been there for many, many, many years, and the biology probably evolves and things change over time. So this is one example here of how you can see that the biology of different lesions can be different. You can see that lesions that are very metabolically active, so very highly avid on FDG and very cold on the NeoB, and others that are very hot on the NeoB and cold on FDG. So there's a lot of heterogeneity in the expression of these tumors. But this is a summary image of the 12 patients. Of all these patients, all of them had, well, 9 of them had what we defined as high tracer uptake. This was defined as having twice the SUV value of the liver. 2 patients had lesions that were less than twice the value of liver, but still showed detectable uptake, and 1 patient was negative. So in this population, at some point in time, we get overexpression of this biomarker. So this is a biomarker that is relevant to GIST, and it's probably something that can be explored. So, summary of this paper, which is, we can announce here that it has been accepted. It's David Pulse's paper has just recently been accepted in EJNNI research, was that in these SDH deficient mutated GIST, we can have very high GRK expression. That expression in these tumors is highly heterogeneous, and this might be related to when we actually study these patients. The levels of uptake are compatible for the use of this as a diagnostics to identify areas of disease. And the uptake level, so in terms of the SUV values, are also comparable with the levels that other agents that we use for theranostics, such as PSMA or somatostatin agents, are seen. So obviously, these are just very nice pictures of tumors, but we need to explore the potential in theranostics. So what is happening with that? So the only existing published example of use of a lutetium labeled agent in patients is this experience from a German group in RM2, where they did a phase 1 dose escalation study in patients with prostate cancer. And here they show that we could get high uptake with decent retention times, showing that you can actually use a lutetium labeled agent to deliver treatment. This was only a phase 1, so they only looked at the toxicity. There's no data still out on sort of longer term effects. So we were involved in a study called NEORA. I'm glad Simon's here. Simon was instrumental in getting this running and operating here. We did treat in this study 2 patients with GIST and I think 3 or 4 with prostate cancer. And here it shows you the things that we do about imaging with the gallium agent, and then we can deliver therapy, which thanks to the physics team here, we deliver downstairs in the nuclear medicine units. We can give lutetium NeoV, and then we can image for multiple time points following treatment to look at retained activity and absorbed dose. So there is a paper in preparation now describing all the findings of this dose escalation study. Is probably going to be out in the next few months, hopefully. But the bottom line is that, as you can see, these images on the right, there's progressive, very rapid release of activity. So while we have a very good imaging tool, yeah, we can see lesions very well, the retained activity here doesn't seem to be enough. So it's back to the drawing board in terms of the therapeutic aspects of this. And so that's something that possibly is an area which we will expand through our collaborations in coming years with other small biotechs that develop these ligands. So I'm going to hand over to Harriet, who's going to talk a bit about the other projects that we have going.
So just a little bit of background to the first study that I'm going to talk about. So monoclonal antibodies have undoubtedly played a key role in the transition from conventional to more personalized medicine. For example, drugs such as antibody drug conjugates, which are designed to directly target the cancer cells, delivering the best therapy with the highest safety margin. Antibodies can also be labeled with radioisotopes, which makes them ideal for PET scanning. And the use of these radio-labeled isotopes allows for the interrogation of specific biomarkers in vivo, providing a one-time diagnostic image in oncology. So often antibodies, because they have a relatively long biological half-life, they can be conjugated with zirconium-89, as this has a relatively long radioactive half-life of 3.27 days. So it's more likely that when the antibody is found its target, there will be radioactivity and we'll be able to see the lesions on PET imaging. So the first study I'm going to talk about is currently open, and we have recruited 4 patients and hope to be a 5th now as well. We're screening next week. So this involves patients with high-grade serous ovarian cancer, and it's a PET study using an antibody to CA125. So we know that high-grade serous ovarian cancer cells very often express a protein called CA125, which is shed into the bloodstream. And on this image here of this histological image, you can see it's stained brown on these ovarian cancer cells. So we have an antibody to CA125, which is currently in therapeutic studies at the moment, called origovimab. And we have conjugated it with zirconium-89 for use as a PET imaging tracer. And this is the first time that this study has been done in humans. So it's a small study of 10 patients, a physiological proof of concept study. So up to 5 of these patients, we aim to give just the antibody on its own first. The idea is in order to try and block any circulating CA125 and hopefully improve the image quality. And then we are imaging these patients at multiple time points over the course of a week, up to 5 time points with PET MRI to try and focus on the best time point for imaging. The preclinical studies were very positive and demonstrated that this tracer can not only visualize the primary tumor, but also the ipsilateral metastatic spread through the lymph node chain in mice. And this was validated with histology as well. So moving on to our first patient that we scanned. So this lady was given the cold unlabeled antibody, followed by the hot labeled antibody. And we learned a lot from this case because it was obviously the first case. So you can see day 0, the tracer has made it into the main vessels. And then by day 1, it started to diffuse into the peripheral tissues a bit more. Day 2, we got a pretty good image of some of her cancerous lesions in the pelvis, in the spleen, and in the pouch of Douglas. And then by day 5, the image quality wasn't as good. There was a bit more noise because the radioactivity was decaying. So focusing on day 2, where we thought we had the best target to background ratio, you can see on the axial fused images, the hot spots in the spleen and in some of the lymph nodes as well. In particular, and this is obviously compared to CT, in particular at the bottom, a lesion which appears very avid, which is quite difficult to see on the CT itself. So patient 1 was pretty successful. We moved on to patient 2, only gave her the hot antibody to see if there was any obvious differences that we were missing. And as you can see, the images actually came out better. This lady imaged on day 3 to day 7 has a very good target to background ratio of her lesions, her disease characterized by metastases in the liver and in the peritoneum and also in the lung. So even by day 7, you're still getting a very good tumor to background ratio. So moving on to some of her axial fused images as well, you can see very avid spots of cancer when compared to the CT scan. And I should mention also that it's probably quite obvious, but the antibody is metabolized in the liver, hence the liver appears quite hot as well in these images and excreted in the bowel. So you can see it in the bowel here as well. So the next patient was given just the hot again. And as you can see, a bit like patient 2, her lesions were very avid from throughout the study, basically from day 3 to day 7. And then the 4th patient, it says 5, but it's the 4th because 1 patient didn't make it through screening. We gave the cold antibody to preload the patient before we gave the radio-labeled antibody. And again, similar to patient 1, you can see the disease. You can see the disease well, but it's maybe not as defined as it is with the patients who just have the hot antibody on its own. So some of the team have been doing some analysis on time activity curves, but this is patient 2 who had the liver lesions. And it's quite interesting to see sort of the SUV max over time of some of these lesions in the liver and in some of the lymph nodes when compared to background organs. And what's really interesting is that we can use this information to predict how much radioactivity is being received by the tumors, but also by the organs. and then estimate from this how much radioactivity would be received by beta emitters or alpha emitters, such as lutetium or actinium, with a view to potentially changing this study into a therapeutic study by using the antibody with therapeutic radiation to deliver to the cancer cells. So yeah, that's particularly interesting as an oncologist. So yeah, to summarize, the next steps would be to finish the study, 10 patients in total, and try and optimize the imaging technique and use this hopefully in the future as an imaging technique that we can order from the clinic to help us localize recurrent disease and add value to existing standard of care imaging. But more exciting, could we take this to a therapeutic strategy where we can treat ovarian cancer with targeted radioactivity? So we're actually going to liaise with the company that made origofimab and talk about potentially going down that road, but also other companies that have other ligands that target CA125 as well. The benefit of that is that some of these other companies have humanized ligands. Unfortunately, origofimab is a mouse antibody, so there is a chance that giving multiple doses to a patient might lead to an allergic reaction. So we've got a bit of work to do, but it's looking exciting at the moment. This study has been a work in progress for quite some time, and there are a lot of people that have been involved. So this slide is just acknowledging the amount of work that's gone into it and who's been involved, throughout the study. I've only sort of joined recently, but I've joined at a very exciting time, that's for sure. So I'm going to talk a bit about a couple of other studies as well, which we don't have any results for yet, but I'm going to just talk about the concept of them. And this one is actually open at the moment, and we are speaking to patients and sending them information at the moment, but we're yet to recruit our first patient. And this is a study using an antibody to CD8 T cells for use in PET imaging. So why would we be interested in CD8 T cells? Well, excuse me, we know that CD8 T cells can kill cancer cells. One of the hallmarks of cancer is it avoids the immune system, it evades it. So it does this in lots of different ways. Listed are a few examples. Less MHC1 on tumor cells. We need MHC1 to provide a positive signal for the CD8 T cells to kill the cancer. Lactic acid and anti-inflammatory cytokines can be secreted, which are hostile to the immune system. And also, the cancer can express more immune checkpoints. Immune checkpoints provide a negative inhibitory signal between CD8 cells and cancer. There are two well-known ones that CD8 T cells express, CTLA4 and PD1, and cancerous cells express PDL1. And when this signal, when these receptors meet, there is an inhibitory signal saying, no, don't kill me, I'm harmless, basically. And over time, T cells that are exposed to cancerous cells that express PDL1 become exhausted, and by that, they express even more of these inhibitory checkpoints. So, checkpoint inhibitors, this slide is just showing why this is important. For certain types of cancer, such as melanoma, checkpoint inhibitors have completely changed the course of treatment for patients. Where years ago, patients wouldn't be alive by 60 months, this Kaplan-Meier curve, which is something we love in oncology because it shows how well treatments can work over time, is showing that in patients with metastatic melanoma, with the use of one single immunotherapy agent, the bottom line, ipilimumab, that 26% of patients are alive, but with two, double the amount of patients are alive at 60 months. So it's proof that immunotherapy works. And the diagram to the side is just showing how ipilimumab and pembrolizumab and PD-L1 inhibitors work, basically just by blocking those checkpoints and preventing those inhibitory signals. So we know it works for some patients, but not all patients, and we don't necessarily know why that is. We also know that immunotherapy causes some very serious long-term side effects in patients. So listed here, it kind of speaks for itself. So we know it can be very toxic as well. So it would be great if we could find a way to predict which patients are going to respond better than others, and hopefully avoid patients that aren't going to respond going through treatment that could be potentially lethal in terms of side effects. There's a lot of evidence in the literature to suggest that the number and density of CD8 T cells in the tumor microenvironment could correlate with outcomes to positive outcomes to immunotherapy. So that's where the CD8 T cell PET comes in. This is actually a mini antibody that's labeled again with zirconium-89, and a company called Imaginab have created this radio tracer, and we are going to use it to image some patients who are undergoing immunotherapy for the first time. Obviously, Imaginab have their own studies going on as well, and their aim is to kind of help prove that this tracer could be used to predict outcomes to immunotherapy in oncology patients. This is just some pictures from their phase 2A studies. They're currently in phase 2B at the moment, but one such patient with metastatic renal cell cancer who had a very lots of avid lesions, lymph nodes and lung lesions on his baseline scan, ended up having a complete response to immunotherapy. And then this was a very small study, there's about 30 patients, but you can see also that patients who had a complete response and a partial response, that's the purple and the blue box, had a slightly higher SUV max when compared to patients who had stable disease or progressive disease. So we are doing the study on patients with head and neck cancer. Currently, patients who have incurable head and neck cancer are offered immunotherapy. If they have a PD-L1 that's positive on their disease, they are offered pembrolizumab, which is a PD-1 inhibitor. If they don't, but they don't respond well to chemotherapy, they're offered a different type of PD-1 inhibitor, nivolumab. So our study is actually imaging these patients who are naive to immunotherapy before and after their first cycle of immunotherapy with a CD8 PET. And we're hoping to obviously gain as much information as we can from the PET scans, but we're also going to be looking at histology, looking at the density of CD8 cells and other immune markers of interest in the tissue, but also some blood tests as well, looking at ctDNA, T cells and cytokines. So hopefully next time I'll be able to present some results. So the next study, which Flavia knows lots about, is Arctic, which is involving a tracer that targets, it's not an antibody, it's a protein, and it targets cell death. Why would we want to know about cell death? Well, the aim of cancer treatment is to kill cancer cells. Lots of tumours still respond really well to chemotherapy, and we hope by the first cycle of chemotherapy that cancer cells are dying, but we can't always tell that from imaging that we use currently. So it would be really wonderful if we could tell after one cycle of treatment whether the cancer is responding to treatment or not. Obviously, we would change the treatment if it wasn't, and avoid any side effects from the treatment as well. So C2AM is a protein that naturally occurs in the body, and it targets phosphatidylserine, which is an extracellular biomarker of cell death. 96% of it is internalized, and then during apoptosis, it transiently becomes externalized. And necrosis, it becomes permanently externalized and accumulates over time if cells are dying. as you can see from the images. C2AM is, we've conjugated with F18 as well. And this is just some preclinical images of mice who had colorectal xenografts between the shoulder blades. And this is 2 hours sped up over time, showing a mouse before treatment. As you can see, the tracer accumulates in the kidneys and is excreted renally into the bladder. And this is after treatment, after 1 cycle of chemotherapy, sped up over 2 hours. And you can see the tumor there lighting up between the shoulder blades of this mouse. Also, some other areas that are lighting up, which we believe are probably also undergoing the side effects of chemotherapy and cell death, such as the biliary tract. So the Arctic study is going to be on patients with breast cancer, and we're going to be looking at patients with HER2 positive or triple negative breast cancer who are due to undergo neoadjuvant chemotherapy prior to having surgery. The reason for this is these tumor types tend to respond well to chemotherapy. The first stage will be looking at 4 patients who haven't undergone any treatment yet, just to image them with this PET tracer for the first time. The second stage will be patients who just had their treatment, and we'll be imaging them and comparing the results with the tracer, the PET MRI. And then the third stage will be comparing before and after for patients that have had their first cycle of chemotherapy with this imaging modality. So hopefully that study will be opening next year, and we'll get some results to show you next time.
Thanks, Harriet. Right, so I'm just going to spend the last few minutes just going over sort of our broader research plans and how this whole issue about developing radioligands for clinical use works. And so there's a lot of areas where we're actually trying to contribute to. So going from the discovery phase all the way to the clinical trials, as we discussed. So this is a very complex process and involves a lot of different expertise, a lot of people, a lot of money, and a lot of time. But I think we're in a position where we are contributing to all these areas. So this is currently all the, I guess, assets on campus that I could summarize that we are putting together to do all of this. I'm going to start from the top left corner, which is the contribution of the Cancer Center. So the Advanced Cancer Imaging theme. So I co-lead that now, and there's a lot of work that Claire and Liz do in project management there. So there's some core funding there that we utilize to sort of drive pilot projects, and the CA125 project, for example, that Harriet talked about, that was funded through the Advanced Cancer Imaging theme. So obviously, being Cancer Research UK, a lot of the sort of basic concepts and the preclinical models and preclinical sort of approaches to these are developed there. The C2AM project, this is a project that Kevin Brindle has developed over many years, and then Flavio and Ahmed more recently have picked up and are doing all the work in terms of trying to bring this now to the clinic. Yeah, CRUK obviously funds also other things that are important to get this sort of these radioligand type research going. Obviously, us here on level 5, the Department of Radiology, we, I guess, coordinate the research, more looking at the clinical side and involved more in image analysis and coordinating the studies. So we have help. Obviously, here we have, for example, Roy, who does a lot of the data analysis. We have Joe, Maria, and Martha have actually been very helpful in coordinating and doing in running these trials when they get to the clinical side. Flavio is working here to develop some more basic radiobiology aspects here on level 4 in the lab. The AMB, the Anne McLaren building, this is a lab in the animal facility that Ferdia set up a few years ago, and now Josh is running, and that's something we're counting on to develop sort of more of the initial animal experiments to prove concepts before we translate these into people. And Flavio and Nisha are working there to sort of implement some of these activities. The Cancer Trial Center, obviously, when we get to the clinical aspects and collaborating with all the oncologists, obviously, you know, we have Simon Pacey was here, but also we're working with Will Ince, the project on Zephyrs that Harriet mentioned. That's with Will Ince for the head and neck cancer, and so there's a lot of industry trials also that proposed through there that we get involved in to test these concept then in clinical trials. The MICL, the Molecular Imaging Chemistry Laboratory, that's part of the WIBIC, that's Franklin Agbirio's reign, mostly developing methods to label with F18 and carbon-11. That's been there forever. It's mostly been focused on neurotracers, but we are making plans to have method development for radiometals, which are more relevant to cancer. And so that's something we are working on. CUH nuclear medicine, where PET-CT and our gamma cameras are placed, and I can't move that thing, but anyway. And all the help from the physics team, Sarah and her team, and Steve and Chess and all the others that are there. We do a lot of work there in getting all these trials and the imaging associated to trials done to get these dosimetry aspects. And obviously the radiographers both in PET and SPECT that help us do the PET imaging and the dosimetry imaging. CUH radiopharmacy has been instrumental in that study on GIST. So at the time, Faxon was actually very helpful in setting that up. And we did most of the study under Vicky Gibson, and Chandra and Fong helped conclude that. That was one of the successes of actually a research tracer that was provided in a kit, which actually could be done in CUH radiopharmacy. I think all the things that will be going forward will mostly be in WIBIC, because that's more complex chemistry in these types of studies. So we require sort of more, I guess, radiochemistry related expertise. And obviously the WIBIC is one of the main areas where I'm concentrating a lot of research now, because there's, again, the drug aspects of this is very important. So the developing the radiochemistry and providing the radiochemistry, and Graham and his team are being very helpful and supportive in how we move this forward and moving beyond the main mission of the WIBIC, which is neurotracers, and getting into doing metals and chemistry related to cancer. Cyclotron, the WIBIC, PET-MR that we're using, because PET-MR is very useful in doing these more complex studies. We have a lot of time on PET-MR. We can do a lot of imaging time points and multiple scanning. For example, the CA-125 requires 50-minute emission scan, which is something that's not very easy to do in a clinical setting where you have a scanner that's continuously needed. So we do a lot of that work there, and Vicky Lupson and her team have helped us with that, and Tim in setting up the protocols and the analysis. So this is all the people that are generally involved in all of these things that we do. And just to go back to the drugs, this is what we have here. We have a lot of capabilities, a lot of things that are happening. The drugs highlighted in red are the ones that are actually clinical. They're sort of NHS funded in some way. And there's a lot of activity going on. The metalmodate and CETOs, an area where Mark Grinnell and his group have done a lot of clinical work on, and HK has helped develop in past years. There's work on methionine that Dan Gillett, again with Mark, and them are working on for pituitary adenomas and also hyperparathyroidism. We image with that. And obviously the gallium dotatate and gallium or F18 PSMA now, the WIBIC RPU is going to be making one of the best analogs available to image F18 PSMA. That's going to be starting in November. It's going to be done in a partnership with Curium, and we're going to distribute that tracer to the region, and we'll also be able to use it here. And then there's all the drugs that we're using there. That's sort of my group that is sort of there, and those are the ones that are actually in use now. And We're planning to expand that by several initiatives that are going on in collaborations with small biotechs, both locally and internationally, and getting funding for PhD studentships. And that's Flavio and Show there in the bottom right. That's the WIBIC RPU, that's Lab 2, which is where all of that stuff, or most 90% of that stuff actually happens. So that's an area that we're developing. Big item for our research in coming years will be this, the long axial field of view or total body PET. That's something that's happening now. It will take another year or so to implement. We have received this funding from the MRC for £5.5 million to set up this well, it's going to be going in the COH PET-CT area. And this effort was largely driven by Franklin, who actually lobbied the MRC to have a total body PET program for the country. And then ultimately resulted in also us being able to, he was the lead in the application for this, and resulted in us being able to have this here. We're going to go over the advantages of total body PET. So it's essentially a scanner that can cover about 106 centimeters. This is the Vision Quadra that we're going to be getting. The advantages are that you can get higher sensitivity because you have a lot more detectors, so you can count a lot more radioactivity. You can potentially reduce patient exposure by significant amounts. You can improve sensitivity statistics. You can do whole body dynamic studies, which is important for research mostly, to do genetic analysis. And you could think of many, many new applications of things that potentially you couldn't do on a standard field of view scanner, just because you didn't have enough time to measure. With total body PET, this makes it all possible. So just to give you a taste of what this can do, this is a whole body scan. This is actually done on a united imaging device. This is a 200 centimeter field of view, first generation scanners, which is actually no longer being produced because it's just way too expensive. But you can do dynamic acquisitions and get whole body distribution. So you can use the same data to either get an SUV image. So this would be the equivalent of what we do for FDG clinical studies, where we do an SUV image at 60 minutes, say, after injection. But you can use that same data and model it to get PATLAC analysis of the KI, or you can do blood volume or K1. So you can test many, many different parameters, much in the same way that we do with certain MR studies, like with the C13 projects that Freddie is following. So this is very exciting in terms of sort of the research aspects, and we can do a lot of characterization of biology and looking at how these tracers work in much more detail with this approach. And just going back to the antibody story, so this is something that I think we will invest a lot on, because as antibodies are at the center of therapy, looking at biodistribution of antibodies in vivo will be something that's really critical to us. We hope to get the technology to label zirconium locally and label, all sorts of molecules of interest. And this is a comparison here of a standard field of view scanner against the use of the Vision Quadra, which is the scanner we're going to be getting. And you can see that the same amount of radioactivity, these are the same patients scanned on the two scanners. You can see this is the quality obtained at 45-minute acquisition with the standard field of view Siemens MCT scanner. You can look here, it's fairly comparable to a 3-minute acquisition with the Vision Quadra. So these sort of applications where you can't inject too much activity, because obviously zirconium has a very long half-life, so you can't inject too high activities because it would be too high exposure for the patient. These will be the applications where this will be very, very important. So the plans that we will focus on are theranostics and getting into trials on that, using total body PET to characterize these tracers initially. Use this for pediatric cancers, obviously having low activity imaging to reduce exposure, and also getting very rapid scanning, so perhaps avoiding the need for anesthesia in pediatric patients. Imaging of yttrium-90, we use PET to image patients that are having CERT, for example. We can use this to do more accurate dosimetry using this approach. Whole body kinetics again, antibodies, as I mentioned, and also potentially doing more low dose surveillance scanning. We have a project to do something like that in melanoma, for example, where patients with melanoma are followed up, and usually very young patients, they have multiple PET studies. If we could do it at very low dose, it would actually be sort of globally reduced exposure significantly. And one of the other things we'd probably be looking at is how to use carbon-11 tracers in whole body applications, which is something that's not done because just the half-life of carbon-11 is too short. But with this type of imaging and equipment and the expertise that there is here in carbon-11, we can do. So just to close, I think we've gone over a bit. So very exciting times for us in oncology. A lot of things happening. Theranostics is a big deal. It's one thing to get very nice pictures of disease, but it's another thing if you can actually use it to treat and cure. So this is something that's really important to us. PET is exploding. We get more and more requests and need for PET and accurate patient management. We can use this to do better tailored therapy, understanding cancer pathophysiology, and use this as a tool for drug development. So that closes, but I just wanted to plug something here, especially for the trainees. If you're interested, the CRK Cancer Center is now advertising these clinical research training fellowships. So these are PhD programs for 3 years, and both me, Ferdia, and other members here up on level 5 have projects that we put there. If anybody's interested to apply, it might be something that is good for your careers and developing research in these areas. And that is it.